Okay, morning. So we're going to start plant transport, and the first thing we're going to have a look at is the different cells, the different tissues, and where you find them in their arrangement in the plant. So this is a giant sequoia, otherwise known as a redwood, and you get these in California, and you can see just how little the people are at the bottom there. So the largest one is 367 feet tall. So we have a problem. So some plants can grow very large indeed. The tallest living plant is a coastal redwood sequoia in California, measuring 367 feet tall. It is a conifer. It needs to take substances from its environment and return wastes to the environment. So pause the PowerPoint and consider the following statements. So every cell of a plant needs a regular supply of water and nutrients. Why is this more of a problem for sequoia than for a daisy plant? Roots can obtain water and dissolved ions. Leaves produce sugars. The reverse cannot happen. What problems arise from this fact? And then how does a plant solve these problems? So have a little think about these questions. Now, just to put something into context, you're going to be studying something called Dicotyledons. Now, there are different types of plant. They fall into dicots and monocots. Now, the arrangement of the xylem and the phloem that we're going to have a look at is only true for dicots, dicotyledonous plants. You don't need to know what a dicot is, but if you come across any pictures, if you're doing some research, you'll see different kind of arrangements. So I thought I would mention it. So cotyledon is what forms the first leaves. Now with dicots, when the plant grows, it sends up a shoot, and if it's a dicot, the first leaves come out in pairs. If it's a monocot, then what happens is they come up one at a time. So most grasses are monocots. The trees, most bushes and shrubs are dicots. So let's have a look at the difference. So you can see here you've got the monocots and then you've got the dicots here. So monocotyledons, they've got one embryonic leaf. Their veins, if you looked at the veins in the leaf, they run parallel to each other. You can't see that. They run parallel to each other through the leaf. So if you look at like a grass, that's what happens. Whereas in a dicot, they have that central mid vein and then they have smaller veins coming off it. That typical leaf shape that you used to draw when you were a kid, that's a dicot leaf. Whereas a grass kind of goes more like that. Okay, now it's really obvious in the leaves, having a look at the veins, and it's very obvious in the stems as well. The vascular bundles, now vascular bundles are phloem and xylem, and in a monocot they are just scattered quite liberally throughout the stem, whereas in a dicot they're arranged in this circle around the edge of the stem. So they're the main differences. They're the ones that you're probably going to be most familiar with. Not many people go around cutting stems, but we're going to look at the stems so you will see those differences. Okay, so how do plants meet their needs? How does the activity and respiration rate of a plant compare to an animal? If you want to pause and have a little think about that. Obviously, animals are going to be more metabolically active. They maintain their body temperature. They are much more physically active. We move around a lot more. And it's not fair to say that plants don't move. Plants do move. They grow towards the sun. Their roots grow towards gravity or grow towards water. And if you've ever seen kind of those little tendril things on a climbing plant, they start by whipping around in big circles until they hit something, and then they start growing around it. So, so you can always Google and watch some videos on plant movement. So they do move, but they don't move as much as animals. That is fair to say. So could the demand for oxygen be met by diffusion? Well, it might surprise you to know that actually it can. They have stomata, and so that's how they can get the gases, get the oxygen, well, get air containing oxygen into the leaves. But they have things called lenticels, and we're going to have a look at that on the next slide because I've got a picture of some. They are a little bit like stomata, but they are on trunks and branches, and they allow for gas exchange directly into those cells. So where is sugar made? You should be able to easily answer that one. But again, have a go at answering it. But they are made in the leaves. So sugar is made during photosynthesis in those palisade cells mainly. Where is it needed? Any ideas? Well, it's needed everywhere. It's needed wherever respiration is needed. So growing tips, growing roots, meristems, stem cells when they're starting to differentiate, growing. If you cut a shoot and it starts to make new roots, it's going to be needed down there. And then sugars can also be moved to storage organs or moved to flowers into nectaries and turned into nectar. They can be stored in seeds. They can be converted into starch and stored in the leaves. So sugar is needed everywhere. Where is the water absorbed? You should all absolutely know this one. It is absorbed through the roots, and we're going to have a look at that in a later lesson. And where is it needed? It's needed for photosynthesis, so it's needed in those palisade cells still, but it's also needed in all cells. Remember, you have a cell wall, and just inside the cell wall you have the cell membrane. Oops. That's supposed to be inside. And then inside that you have the vacuole. Now if water is taken into the vacuole, that vacuole swells up. Let's try and swell our vacuole. And what happens is it pushes that membrane, that cell membrane, out to the cell wall. And so you get something called turgor pressure. Turgor pressure, and that's what helps to keep the plant upright. So that turgor pressure is really very important, and if you don't water your plant, then that let's see if I can get rid of this. Let's get rid of that as well. Then that cell wall will start to move. Oh, hang on, let's make that better. If you don't water your plant, then that vacuole will shrivel and your cell membrane might move away from your cell wall, not giving you that turgor pressure, and your plant will wilt. Okay, so do plants require a transport system? Absolutely. Now if you've got, so plants come in all shapes and sizes. If you've got something that's very, very small, you can rely on having much simpler systems, but something like that giant sequoia, it's going to need a lot of xylem and phloem. It's going to need a lot of support tissues as well. But it absolutely needs a transport system to get it from potentially a couple of 100 feet below ground, and then 367 feet up to the leaves right at the very, very top of the tree. The roots of a tree quite often mirror, not necessarily as deep as they are high, but they mirror the shape of the tree. So those roots will be going down quite a way into the ground. So it's not just trying to move the water 367 feet. Those roots go quite a way into the ground, so it's probably nearer 400-500 feet. Okay, so you've got this gap fill in your book if you want to have a go at filling it in. So if you want to pause the video, and then we'll have a look. So water is moved up the plant from the roots in xylem tissue. And we're going to look at the difference between xylem vessels, which are cells, and phloem tissue. Sugars are moved up and down the plant from the leaves or storage organs actually in the phloem tissue. Okay, right, so you've got this diagram in your book and you can annotate it to show all of these different bits. So please look at this bit as well. So we're going to have a look at these lenticels. So these are in woody trunks and branches and help gas exchange in the rest of the plant. So this is what they look like. These are those lenticels here. These little things here are the lenticels, and they allow air containing oxygen to get into these cells for respiration. Okay, and then obviously on the bottom of the leaves you have the guard cells. Well, that's the stomata, and these are the guard cells which allow it to open, or the guard cells control whether the stomata is kind of there or not. So the movement of the stomata opens and closes. The movement of the guard cells, sorry, opens and closes the stomata. So now we're going to focus on these things. This is a vascular bundle. You've got xylem and phloem tissues make up a vascular bundle. Okay. So this is the arrangement of vascular bundles. Vascular bundles. That's supposed to be an R. Bundles. Oops. Vascular bundles, and this is a dicot arrangement. So dicotyledon. So we've got the leaf, the stem, and the root. Now in the leaf, you've got the xylem on the top, and you've got the phloem on the bottom. Now in the stem, the xylem are on the inside. You can see them just here, and the phloem are on the outside. Now, a way to try and remember that maybe is if you can remember the leaf, and then if you rolled up the leaf, the xylem would be on the outside, sorry, the phloem would be on the outside, and the xylem would be on the inside. It's up to you, but you need to remember. Now for your exam, they prefer more like 5 vascular bundles. So a vascular bundle is the phloem and the xylem. Okay, that is a vascular bundle, and you find them in the stems. So these arrangements, so like I say, you probably, if you're asked to draw one, I would probably draw 5 or more. And so you get them around the edge in kind of like a circle. Okay, now in the root, your xylem is in the middle and forms either an X shape, or sometimes you can get more than one. So it kind of looks like the spokes of a bike maybe, and the phloem are on kind of in the middle between the xylems. Okay, now something to note is that your xylem and phloem should take up less than half of the overall width of your root. So if you were drawing one, let me just get rid of this stuff. If you were drawing one, you would do that for the outside of the root, and then you would do your xylem in the middle with your phloem in between. Okay, but you can see it shouldn't be this big thing. It doesn't take up all of that root. Okay, right, so this is just showing the same thing again. So you can see the leaf here, the stem, and the root. Now this has included something called sclerenchyma. Sclerenchyma, doesn't matter how you pronounce it. Now sclerenchyma is not part of the vascular bundle. It is a support tissue. So you find it on the outside of the phloem, not in all plants, but it is there for support. Because obviously if you get tall plants, the xylem is lignified, so it is quite sturdy and strong, and it is there for support as well as transport of water. But sometimes they need a little bit of extra help. So sclerenchyma is also impregnated to give it that rigidity, to make it a support tissue. Okay, now you can see on the root the xylem in the middle with the phloem, and then we're going to have a look at something called the endodermis later. So you've got the xylem, the phloem, you've got a layer of cells called the pericycle, and then on the outside of that you've got a layer of the cells, oops, not epidermis, sorry. The epidermis is the outside, the very outside, sorry, the endodermis. We're going to have a look at that later because that's very important in the transport of water into the xylem from the root. Okay, so epidermis down here is just kind of this outside part of your root. Very sorry about that. Right, so just more pictures. This one is obviously of a real plant, so you can see the xylem in the middle. Now this has used a differential stern, so you can pick out the xylem from the phloem. It is very, very, very difficult to then pick out the endodermis and the pericycle layer. You have to be you have to really know what you're doing to pick out those. I'm not kind of good enough to pick out those usually. Okay. And again, you can see these vascular bundles forming this kind of circle around the edge. Now, something that you can see here is, you can't see the labels, but that would be the xylem. That's not a very good arrow. Let's get rid of that. So that would be the xylem. That lighter green, ugh, I haven't done it very well. This is rubbish. Remember, I'm trying to do this with my fingers. This kind of lighter green colour here, that is the phloem. You've got cambium as well. And then this pink stuff, pinky, reddy, browny colour, that's the sclerenchyma. So this arrow here is pointing to the sclerenchyma. This arrow here is pointing to the xylem, and then this one is pointing to the phloem. Okay, so the sclerenchyma isn't part of the vascular bundle. You've got the xylem and the phloem with cambium in the middle, and you can see here you've got sclerenchyma labelled as well. Now parenchyma is all of this stuff in the middle. It's just kind of like a packing tissue, so it's just filler really, packing. Now this arrow here, let me try and highlight it in a different colour. Let's get rid of these for a second. This arrow here is pointing to another support tissue called collenchyma. Colon, and then it's just the chyma stuff again. And that's another support tissue. And you are going to be writing some notes in your book from the textbook on these different tissues. Right, just another one. So you can see this is the vascular bundle. It does not include sclerenchyma. Okay. Right, and the leaves. Now this is where it gets quite tricky to pick out the phloem from everything else, but you can see the xylem here, and you can see the phloem around there, and this is that spongy mesophyll, and down here you've got stomata and guard cells. This would be the top of the leaf, and this is your palisade layer where you get most of your photosynthesis. Right, so we would have done an experiment at school where we would have showed you where these vascular bundles are in celery. If you're not self-isolating and you can get to a supermarket and get some food dye and some celery, you can have a go. So what we would have done is we would have put some food dye into some water and put some celery in. Now there is a difference between dye and stain. Dye doesn't really have the ability to kind of penetrate into the cells. It doesn't stain differentially, and so it's just there to see the position. So what we would have done is we would have put the celery in the water, and then to show you where they were, we would have done a transverse section. Now a transverse section is cutting across that way, and then you can see these dots. These are vascular bundles going up the stem, each of these. Okay, and the same on this one. All of these big dot things are the vascular bundles. Okay, so you take a section, so you get a nice clean cut through, and you'll be able to see those. Or what you would have been doing next lesson is a PAG, and I will still upload that PowerPoint so you can have a look at what you would have been doing, and we will ask you to do it when we come back. What you can do is cut a section, so you would have just not put it in die, just taken some celery. So we'll just take some celery and you would take a transverse section. Now you would try to get that transverse section as thin as possible, and then what you would do is stain it, and you can stain it with something called toluidine, oops, that's an E, blue. Toluidine blue stains lignin, so you can see the difference between the xylem and the phloem. So you would take a very thin transverse section, pop it onto a microscope, or well actually you stain it in a dish first. So you stain it with toluidine blue, and then you'd be able to look at it under the microscope. Now you look at it under lower power. If you go too high, because you can't make a thin enough section, you just see blurs. So you stain it with toluidine blue, and you look at it on lower power, and you'll be able to see the vascular bundles. And in the next PowerPoint presentation, I'll show you some of those slides. Right, so your PAG would have been next lesson. So for the rest of this particular lesson in your workbook, you need to fill in the pages on the different tissues, and they are all in the textbook, so you can easily have a look at those. Okay, thanks.